So first of all, thanks a lot to the organizer, to David and Ran for inviting me. I see a lot of familiar faces in the audience, and that's great. So I'm looking forward to the discussion that we're going to have after my presentation. So you see the title is in a form of a question, and it's intentionally provocative. And it requires some explanation, and I'll give you this explanation in the next slides. But essentially, the main idea is that our hypothesis is that the hippocampus is primarily a memory device. And when it is exposed to the statistics of memories that you want to memorize where you're navigating in an environment, then you get response properties that are very similar to those that have been recorded in the hippocampus during navigation, like place cells. So I have a probably yes to the as a response to the answer, because we have some preliminary data that James Priestley collected in Attila Losonczy's lab. They're still preliminary, but we are very excited about them, and they really show how we want to prove that our ideas are correct, and so it will be interesting to discuss them. Okay, so let me start by thanking all the people who were involved in the project. It all started with Marcus Benna, is now a professor at UCSD. He's recruiting. If you're interested in this project, I know that he's working on that. And then James Priestley and Attila Losonczy got interested in the theory, and they designed an experiment together with me to test some of these ideas. And I'll tell you a bit about that towards the end. And finally, Lorenzo Posani, who is now in Paris, temporarily, is trying to get his visa, and then hopefully will come back to Colombia, is working on the theory. Okay, so if you want to read more, we already have a paper about the theory. It's in PNAS. As you can see, the journal did not allow to have a question mark in the title, but it's basically the same content. And there are all the details about the theory and much more than what I will be able to tell you today. Okay, so it all started a few years ago. There was one of these annual events at the Rockefeller University where they celebrate the centenary of a famous neuroscientist. Now, usually it's in memory of the neuroscientist, but in the case of Brenda Milner, she's still alive, and she's 104 now. And at that time, she was turning 100. And I guess you all know that she's famous for her contributions to the understanding of the role of the hippocampus in memory consolidation, in particular for her studies on patient AHL. So, there is a huge literature, especially on humans, that focuses on the role of the hippocampus in memory. Now, at that workshop, John O'Keefe was present, and he gave a presentation, and he tried to put his studies in a historical perspective. And he said that he was very much inspired by the work of Brenda Milner. So when he started to record from the hippocampus, he was really looking for memory cells. And then instead, he found these place cells. Now, what I will try to convince you today is that actually these are the same cells, and they're probably important for memory. And we really believe that hippocampus is fundamentally important for memory consolidation. Okay, so let me start from the memory aspect of this presentation. And I will start immediately with this paper that came out in 1982 that attracted so many physicists to neuroscience. And it's a memory about associative memory. And John Hopfield came up with a very simple prescription to find the synaptic weights of a recurrent neural network in such a way that you can store a very large number of memories. So what does that mean? I mean, in order to say something about memory capacity, you need to make an assumption about the nature of the memories, whether they're similar to each other, whether they're structured or unstructured. And John Hopfield, for convenience, he assumed that the memories were random and uncorrelated. Now, this is not a totally unreasonable assumption, despite the fact that we all know that in the real world, memories are highly structured, and so they're definitely correlated. And the reason why it's not totally unreasonable is that the optimal strategy for storing memories is to start from the sensory experience, combine it with what you already know, extract the uncorrelated component, or if you prefer, a compressed representation, where essentially the memories that you want to store are approximately uncorrelated, and then store them in a recurrent neural network or in the network that is downstream of the hippocampus. So assuming that the representations are random and uncorrelated is probably hypothesizing implicitly that we have this kind of machinery that allows to compress the information. Okay, so this is another simple example of compression. Here, I'm illustrating one of the reasons why we can have this seminar on the internet, which is the MPEG algorithm that compresses videos. And here I have, a series of different frames where there's somebody speaking, and as you can easily realize, all these different frames are highly correlated to each other because most of the pixels are actually the same. Oh, we have an alarm in our building. I hope they're not going to evacuate, but let's see. Yeah, today is really a complicated day. Okay, so you see that we have all these different frames that are highly correlated to each other. So what you can do is to transmit or store on your hard drive what is called the keyframe, the first one that I have here, and then just focus on the differences between this frame and the previous one. And as you can see from this very simple example, you obtain new representations here that are compressed for two reasons. The first one is that they're much sparser than the original one. I don't need to store all the information about every pixel. The background is almost always the same. And the second one is that they're definitely less correlated than the original ones. So, I get sparseness and decorrelation. Now, let's see whether we can use similar principles also in neural networks. Okay, so in order to set up a problem that we can study, we wanted to introduce a tiny bit of reality in the memories that we are going to consider for our benchmark. And we wanted them to be correlated. So, now I'm going back to the 80s and the 90s, and there were several studies on memory capacity where investigators assumed that these memories had some structure and they were organized in an ultrametric tree. Actually, here the ultrametricity property is a kind of irrelevant, but yeah, let me explain just what I mean by this ultrametric memory. So you typically start from one memory. Let's say this would be the representation, and this is what I would call the ancestor. So let's say half of the neurons are active and half of the neurons are inactive, and I sort them in such a way that I first have the active neurons, the yellow ones, and then the inactive ones. Okay, so now I'm going to generate a certain number of descendants that bear some similarity with their ancestor. So I just go over this vector here, and I decide with some probability to flip the state of activation of the neuron. So for instance, all these are the same, and this would have different state of activation, and this one also would be different. I can iterate this process as many times as I want, and I can generate a certain number of descendants. Okay, so they're represented here in this graph. All these are descendants. They're all similar to their ancestors, and they're all similar to each other. In other words, they're correlated. Okay, so the situation we are going to consider, and it's really the simplest one we can imagine, is one in which we have a certain number of ancestors, and they're random and uncorrelated here at the top, P ancestors, and I'm going to consider K descendants for each ancestor. So they're all correlated to each other, and they're correlated to their ancestor, but they're not correlated to all the descendants that come from different ancestors. Okay, so now let's try to use a strategy in which I compress the information by hand. Okay, so this is not what I'm going to use. It's not going to be the general model that then we will use for the hippocampus. It's just to give you an idea of what we can do with this representation to construct other representations, to recode the representations in such a way that are more suitable for memory storage. Okay, so let's say I'm starting from this ancestor, and here I hold these descendants. And then I have a second ancestor that is actually orthogonal, in this case, to the first ancestor, and a bunch of descendants. So what I could do is to use a relatively small number of neurons that are encoding just the ancestor. And you don't need that many, because we are assuming that the world is structured enough that we don't have that many ancestors, but we have a lot of descendants per ancestor. And then I'm going to encode the difference between the ancestor and the descendant. So for instance, for this particular descendant here, I have here the code for the ancestor it comes from. And then here I'm activating only 2 neurons, basically, that represent the differences between the descendant and the ancestor. I can iterate this process for every descendant, and I get some new representations. Now, I'm using, in this particular example, more neurons than what I had initially, but I'm gaining something, which is the fact that now these representations are sparse. They're really sparse. Of course, it depends on the similarity between each descendant and the ancestor. And the second thing that I achieved is that they are approximately decorrelated. They're not perfectly decorrelated, but they're certainly much more decorrelated than the original patterns. Okay, so, if you go back to the 80s and the 90s, there were a lot of studies from Michel-Sodix and Feigelman, 1988, Alessandro Treves, that showed that when you have sparse representations and they are uncorrelated, then the number of memories you can store scales like the number of neurons divided by F, which is the coding level, the fraction of active neurons. So if you have very sparse representations, F goes to 0, and you can greatly increase the memory capacity. Okay, so we have a few calculations in the paper that show that this would be the case when you construct these compressed representations. But what can we do in the most general case? Well, if you come with a different example, then can we deal with that? So one general way to construct these compressed representations in a neural network is to use a very old concept from neural network theory, which is the concept of an autoencoder. So you start from an input here, you're going to have some intermediate layer, and here you're trying to reconstruct the input. So in this particular case, I'm considering a situation in which the number of neurons in the input is smaller than the number of neurons in the compressed representation. I want to imitate essentially what happens in the hippocampus, and I will show you later why. So in order to compress the representation, I need to impose something. I need to have some kind of information bottleneck. And so I'm going to impose sparseness. And again, this is another old idea from the 90s. Olshausen and Field published a series of really interesting papers that showed that this kind of sparse autoencoder can reproduce many response properties of the V1 neurons. Okay, so now this is a very general way of constructing a compressed representation. I just tune these weights in such a way that I can reconstruct the input, and I impose sparseness in the intermediate layer. So let's say I play this game with these ultrametric memories, and I construct these representations, and now I can study these representations in many different ways. So first of all, I want to know what happens in terms of correlations. So let's say now I consider these ultrametric memories, and I focus on 2 descendants that come from the same ancestor. And I'm going to change the environment. So I'm going from an environment where I have 8 total memories in this simple example that come from 4 descendants. And I'm going to keep the number of memories the same, but change the level of compressibility by changing the number of ancestors. So here I have many more representations that are correlated to each other than in this case here on the left. Okay. So on the x-axis, I'm changing the branching ratio, but I'm keeping the total number of memories constant. Now, the similarity between each ancestor and the descendant is always the same. So this is expressed by the fact that also the similarity between two descendants is always the same, no matter what is the branching ratio. But when I consider the statistics of the environment and I train my autoencoder, the autoencoder cares about this because the level of compressibility is different in all these different situations. And so you see here that as you increase the branching ratio or the compressibility of the environment, the autoencoder progressively decorrelates more the representations. Now, what can we do with these representations? So, this is what is probably related to what people call pattern separation that has been observed in the dentate gyros, which is the first part of the hippocampus that receives input from entorhinal cortex. And it's probably similar in many ways, and I'll come back to this later. But once we have this pattern separation, what can we do with these representations? So what I really want to show you is that you can increase the memory capacity of the system. And it's not a trivial statement because you need all this additional machinery here. I need this intermediate layer. I need what I will call probably hippocampus at some point during this presentation. But for now, it's just this intermediate layer. And so I need many more synapses in principle. So now I really want to do a fair comparison. I'm considering this neural circuit where I have a certain number of ultrametric memories, and I want to see at a later time when I use a very noisy cue, whether I can reconstruct the input or not. So I have a total of 4 n squared synapses in this case. Now, I'm considering now the other case in which I have n neurons in the input and just n neurons in the output. So no intermediate layer. So I gain a factor of 4 in this case because I don't have all this additional machinery. And for completeness, I'm going to consider also random projections because this has been studied widely in a lot of papers on the role of the hippocampus in memory consolidation. Okay, so now you can run the simulations, and I don't really have time to go into details, but this plot summarizes the results that show that the autoencoder can store many more memories that are represented on the y-axis than when you read out directly the input or you're considering random projections. So this is plotted as a function of the total number of resources, of memory resources, which is the total number of synapses. So we believe it's a fair comparison. And despite the fact that we have this additional machinery, you see that actually even the scaling properties are better than in the case in which we are reading out directly from the input. Okay, so this pattern separation, this decorrelation is useful for something. It's useful for memory capacity. Now, let me summarize a bit the literature and, this idea that, as I told you, is really as old as neural network theory. So let's say I have my sensory experience and I want to combine it with some information about recent experiences, let's say the statistics of recent experiences. So in this case, I'm really focusing on the hippocampus. And our hypothesis is that it's really the hippocampus that is generating these compressed representations. Okay, this idea that the hippocampus is recoding the sensory inputs is really old. It dates back to David Marr's work from 1971. And then there is a series of beautiful papers by Jane McClelland and colleagues. And they all show that actually you can probably use this kind of recoding to generate more efficient representations. But more recently, people thought also about the idea of compressing the information in a smarter way, so taking into account the statistics of the environment. And in particular, Gluck and Myers in 1993, they really hypothesized that the hippocampus is compressing the information in a very similar way to what I presented to you. And then there is a series of other papers, more recent, where they studied the hippocampus in a very similar way, and they showed that also some similar results. Although, none of them estimated the memory capacity. They always assumed that these new representations would be better. Okay, so now let's switch to navigation. Let's say you have your little mouse here in this square arena, And the mouse is moving around. It's exploring the environment. So let's say this is a trajectory. So the mouse goes through a series of sensory experiences. And when it goes through the same location twice, it's going to have two different experiences that of course are different, but they will be correlated. And actually, you know, in any realistic environment, most of these representations of the sensory experience will be correlated to each other. So let's say we want to take advantage of these correlations to generate representations that are more suitable for memory. So we play the same game. We have this autoencoder. We have the inputs here, the compressed representation in the intermediate layer, and then the reconstructed input. Now, I don't have time to go over the details. You can read the paper, but you know, we use some reasonable input that come from entorhinal cortex. We have some implicit information about the position of the animal that is simulated. They're compatible with the current literature, but the kind of representations we have are actually highly entangled, and they're very high dimensional. You know, let's say something like the visual scene that you see when you're walking in a particular environment. So, you know, it's not trivial for the system to extract the information, let's say, about latent variables like the position of the animal. Okay, so now let's say the animal is exploring this environment. I'm considering only this straight trajectories. It's not so important actually, but the simulations that I'm going to show you are based on this assumption. So if I do this, and now I look at the representations in the intermediate layer, I see this beautiful place cell. So each of these square is a place field of the different cells, so cell number 1, cell number 2, and so on. And basically, you know, if I look at one of these representations, this says that this neuron is active when the animal is exploring this part of the environment. Okay? You see a lot of variability between these different cells, and they have many response properties that are similar to what is observed in the experiments. Okay, here, the only assumption we are making is that this system is compressing the information. We are not making any other assumption. We get these place fields. Now, if you use random projections, and again, you use this kind of high dimensional inputs, you're not going to get these nice fields. So this is the best that we could get. And there are many good reasons for this. And if you want, then later we can discuss them. You know, basically, it's really important to learn from the statistics of the environment if you really want to have this kind of place fields. Now, if you have simpler inputs, like, you know, Gaussian inputs that encode directly the position, yes, you can do a good job with random projections, but in a sense, you're moving the problem to an earlier stage. Okay, so now these place cells are not really place cells. I mean, this system has not been designed to encode the position of the animal. It has been designed to encode memories in an efficient way. So let's say that now my simulated animal is going to explore the same environment, exactly the same environment, but with a different statistics of exploration. So it's spending more time here on this left, the upper left corner. So in this case, what we get is a series of different memories. Of course, they will be similar to some extent to those experienced in environment A, but they're different. And this will be reflected by the response properties of the neurons. So you can see here that many of these place cells are similar. Some of them are significantly different. And you can actually look at the fluctuations of the place field to decode the recent history, because I'm storing memories and I'm continuously updating the representations. So what we expect is that these place fields are not stable because we are always memorizing something new. And this is what happens also in the real brain. I mean, you know, typically when you look at the place field, you're just looking at the average activity over 10 minutes of recordings. But these kind of plots don't say anything about the fluctuations, which are actually really wild. And you know, the work of André Fenton really studied these fluctuations in greater detail than anyone else. And it's pretty clear that you have a lot of instability. Now, despite this instability, thanks to the fact that in the end we are encoding the same environment, we can decode the position. So we can use these representations to say something about the position of the area. Okay, so now it's time to show you what kind of evidence we have that this is not totally unreasonable. And the idea comes from this plot that I showed you previously, where you have that the autoencoder decorrelates these representations in a different way depending on the compressibility of the environment. Now, we want to play the same game in a navigational task. And we want to show that in that case, by changing the compressibility of the environment, we are going to get the kind of representations that are predicted by the theory. Okay, so the idea is illustrated in this very simple example. So, let's say you're walking in a desert. It's pretty boring. This is a compressible situation because there is not much that is happening. I mean, most of these stimuli don't really have a meaning for us. They're probably filtered out even before they get into the hippocampus. Whatever gets into there is going to have an autocorrelation function that has a very long tail, because I move 50 meters ahead and I don't see a big difference. Okay? Now, if I'm in a city center, this is in Florence, where I was born a long time ago, and I play the same game. I compute the autocorrelation function. I don't have this long tail any longer. Things are changing here much more rapidly. I'm moving 50 meters ahead. I see a different shop. I see people there. There are a lot of landmarks. And so here, this is definitely a less compressible situation. I have much more information that I can potentially store that might be then relevant for performing a task. Okay, notice that if the hippocampus is really designed just to encode the position and just want essentially a GPS, then you don't expect a difference in these two situations. The GPS would work exactly the same way in these two situations, with probably the only difference that when you're in the city center, maybe you don't get the satellites as well as in the desert. But if it works, it's going to work exactly the same way. Okay, so this is what we are doing with our mice. And so again, this is James Priestley, who did this experiment in Attila Losonczy's lab here at Columbia. Now, James is at EPFL, he started his own lab, and is continuing to work on this project. So you can see this little mouse here. Okay, there is a microscope here because they used calcium imaging to record the activity. And the animal is running on this wheel and it's controlling this virtual environment. So the great thing of a virtual environment is that we can change whatever we want. And in particular, we can control the compressibility of the environment. Okay, so these are the recordings. This is another video. It's they used GCamp8M, and it's pretty amazing. I mean, you can get easily between 3,000 and 5,000 neurons per recording. So you're going to see really smooth curves, and they are not simulations, they are real data, and it's thanks to the fact that we have so many neurons. Unfortunately, we have only 2 animals. That's why I'm saying it's preliminary. But given the number of neurons, I think we can already say something about what is happening in the hippocampus. Okay, so now what I'm going to do in the next few slides is to show what happens in the experiments and what is predicted by the theory. Every time I have a black background, that's the experiment. Every time I have a white background, that's the theory. Okay, so for the experiments, we use these 2 different virtual environments. So basically, in the first version of the experiment, we were changing the gain. We realized it was not the right way of changing the autocorrelation function for several reasons. So we are not doing it any longer. We are perfectly aware that there are issues, but the results I'm going to show you are actually based on this trick. So that basically, effectively we stretch this environment by changing the gain factor. And this environment then turned out to be more compressible than this one here. Okay, so this would be like the city center, and this would be more similar to the desert. Okay, so the first thing that I want to show you is that the animals are not confused by the fact that we are changing the gain. They really think it's a different environment, and they're running exactly at the same speed. So the physical velocity is the same in the two environments. So it's almost indistinguishable. Now, what do we expect in terms of recordings? So let's say that we are in context B that is highly compressible. Okay, so this is the desert. So this is the input that we probably have. Of course, we cannot measure it, but it's some kind of curve that is reflecting the intrinsic dimensionality of the environment that is only one, but it's embedded in a very high dimensional space because we have this visual input that is rather complex. Okay, if I want to compress this manifold, sorry, first let me show you what happens in the low compressibility case. I still have a 1D environment, but now I have all these twists and turns that change much more rapidly. So this would give you a different autocorrelation function. And it's still the intrinsic dimensionality would be 1 also in this case. Okay, if I want to compress this environment and I'm using this sparse autoencoders, essentially what I get is a sort of a local reference frame for a portion of this manifold. So here, for instance, I would have this local reference frame that would correspond to one place cell. And here I would have a second one. And I need a number of these cells that is as large as the number of twists and turns of this manifold if I want to have a good description of what happens. So in this case, then, for the low compressibility, even if I consider the same distance, I will need more of these local reference frames. So in other words, what I expect for the place fields is that the density of place fields would be higher in the low compressibility case and smaller in the high compressibility case. Moreover, the width of the place field should be different, and it should reflect the way the environment is changing. Okay? So here I want to have narrow place fields, and here I want to have larger place fields. Okay, so this is one of the two things that I mentioned. So this is the field width as predicted by the theory when I change the compressibility. Of course, in the simulations, we can have 10 different levels of compressibility, not in the experiment where we have only 2 different environments with 2 levels of compressibility. So the purple one is low compressibility and the yellow one is high compressibility. And of course, there is a lot of diversity across different neurons, and this is the distribution. You clearly see that the 2 distributions are significantly different. And in context A, in the case that you have low compressibility, the place field is definitely narrower than in the other case. This is true also for the density of place fields, and it's shown both for the theory and for the experiments. So in the case of low compressibility, the coding density, the number of place cells per meter, is higher than in the case of high compressibility. Okay, so this is something that has been observed in other experiments, and it's more or less known. But one of the things that we could do with this data was to measure the decorrelation that happens during the learning process. So what we did was to consider one environment, and we did it for both environments. So let's consider one here. And we consider 2 different locations of the animal. So, we have a snapshot of the activity when the animal is at this location, and another snapshot of the activity when the animal is at this location here. And then we just measure the correlation between these 2 representations. Of course, we extract the activity of individual neurons, we get a big vector, and then we compute the correlation. And we measured it as a function of the distance between these 2 locations. Okay, so now what you expect from the theory is depicted here, again, for many different levels of compressibility. And you have that for short distances, you actually want to have a larger correlation, a positive correlation. And that actually helps a lot for generalization. We are always operating in a regime where we have a lot of noise. Okay, so that's important. And then if the distance is large enough, you're going to have these negative correlations. If you go to even larger distances, I'm not showing here, then it will be 0 again, because for larger distances, the representations are decorrelated from the very beginning. Okay, so this is what happens in the real data. So, you know, these are the smooth curves I was talking about. So again, purple line is for low compressibility case. And you see it's very similar to the purple line that I have here. And for the yellow line, I'm in the high compressibility case. And I see that here the tail is very different. Okay, so now you might say, well, you're just reflecting the structure of the environment. Maybe it's already in the inputs of the hippocampus. But this is not the case because we don't have this kind of structure at the beginning. So this is really the critical plot for us, because here we are plotting the change in the correlation as a function of the distance between two locations. And this is, again, for low compressibility, and this is for high compressibility. And as I mentioned, for short distances, you want to correlate even more the representations of two neighboring locations. But then you want to decorrelate. This is the negative part. And then it would go again to 0. It's not shown for high compressibility, it will be for larger distances. So this is what happens in the real data. Again, it's preliminary. It's only 2 animals. We're not using exactly the environment that we wanted, but you know, it's work in progress. And James is collecting more data. And you see again that qualitatively it's very similar. So for the low compressibility case, you have this positive part and then the negative part that corresponds to the decorrelation. And again, for high compressibility, and everything is stretched. It's reflecting the statistics of the environment. Okay, so let me conclude. Correlated memories can be stored more efficiently by compressing the representations. This is not too surprising. We showed it in at least in this simple case of ultrametric memories. And if you're thinking about the hippocampus, it's very possible that the dentate gyrus is involved in this process of decorrelation. This would be compatible with the numerous results that show that there is some kind of pattern separation happening in the dentate gyrus. Okay, compression leads to this spatial tuning. Okay, so we are talking about a memory device that is just compressing the information in such a way that can be stored more efficiently. This spatial tuning emerges very naturally if you make this simple assumption. Of course, now we are not really going to have neurons that encode just the position of the animal. Under some conditions, they would encode the position of the animal. But even under these conditions, you're going to have this history dependence. And you certainly expect them to be highly variable in time. Now, not only they are highly variable in time for subtle changes like the statistics of exploration, but whatever happens in the brain is going to change them. And this is a point I'm going to make them later, but it's something to keep in mind. Now, despite all this variability, they are stable enough that they allow for decoding the position of the brain. Now, the other prediction is that the hippocampus would compress any type of memory, whether it's spatial or non-spatial. And that's something that can be seen in a lot of studies. In particular, Dmitry Aronov showed that you get this kind of place field even in the auditory space when you present stimuli using a statistics that is similar to what you would experience in a navigation task. Okay, so this is something I already mentioned. You have a lot of variability, and the representations are going to be affected by whatever happens in the environment, whether it's salient or non-salient. But salient events like a delivery of reward are going to change the representations in a more dramatic way. And that's certainly observed in a number of experiments. And we also reproduced it in our paper, so you can find it in the supplementary information. But what is called remapping should probably be interpreted in a different way. It's just reflecting the storage of new memories. Okay, so finally, I showed you that we have some preliminary experimental data that is compatible with the predictions of the theory. We are perfectly aware that these studies are not conclusive. They're alternative interpretations, but they are certainly all incredibly compatible with the predictions of the theory. And so we are encouraged to continue. And if you have any suggestion for other experiments that would really reveal some signature of this compression process, please let me know. Okay, so I went a little faster because we started a little later, but yeah, but I will stop here, and I'm happy to take questions.